remarkable finding of genetic studies in recent years has been that at the beginning of life on Earth, when the tree of life was starting to grow, there was quite a lot of horizontal or lateral gene transfer. Horizontal gene transfer means that genes can be shared not by common ancestry, but via a direct transfer mechanism between different species. For example, scientists have found a bacterial gene in the genome of some jellyfish in the structure that forms the sting of the jellyfish. Since this gene is not present in any of the ancestors of the jellyfish, but it is found in all bacterial genomes studied, it is thought to have been transferred horizontally, directly from bacteria to jellyfish. And the same happens with many genes of a bacterium called Wolbachia, which parasites the eggs of flies and wasps. Scientists have recently found up to 1 megabase of Wolbachia genome in the genome of one species of Drosophila, as well as in the genomes of other worms and wasps. This kind of horizontal gene transfer apparently was very frequent at the beginning of cellular life on Earth, and this contributed a lot to the reshuffling of early genomes. So during the time of Luca and the formation of the first branches of life, there was a lot of horizontal gene transfer, mostly via small DNA fragments that entered into cells, a process known as transformation. And viruses also are thought to have played a very important role in this process. In fact, some researchers talk about two empires of life: viruses, non-cellular organisms on one hand, and cellular life forms comprising the other empire with its three domains. Horizontal gene transfer was so prevalent at the very beginning of cellular life that scientists today talk not of a tree of life, one tree of life, but a forest of life, and not one Luca, but many Lucas. And then within this forest, it is thought that endosymbiosis of a proteobacterium, either by an archaean or by an eukaryotic ancestor, led to the first eukaryotic cells. Many of the genes of the engulfed bacterium were transferred to the genome of the eukaryotic host, and this finally led to the typical eukaryotic cells, which have a nucleus, a system of internal membranes, and mitochondria. We know that this process was completed at least 2.1 billion years ago, when we find the first fossil of a eukaryote called Gripania. But of course, it could have started much earlier. In fact, recent genomic research has unveiled what might have happened to early archaean genomes around 3 billion years ago. In a paper published at the beginning of 2011, scientists traced thousands of genes from 100 modern genomes back to the moment when those genes first appeared on Earth. The work suggests that the collective genome of all life underwent an expansion between 2,800 million years ago, during which time almost 30% of all gene families existing today came into existence. And this is known as the Archean expansion, which then tailed off and was progressively replaced by gene duplication as the source of new genes. The authors believe that the cause of this expansion was the birth of the modern electron transport system, the biochemical process by which electrons are moved within mitochondria and chloroplasts during the production of energy. The evolution of electron transport during this Archean expansion would have enabled several key stages in the history of life, including respiration, photosynthesis, both of which could lead to much larger amounts of energy being harvested and stored in the biosphere, and an increase in atmospheric oxygen concentration. This study also suggests that oxygen-utilizing genes did not appear until the tail end of the Archean expansion about 2.8 billion years ago, which is consistent with the date that geochemists assign to the Great Oxidation event about 2,400 to 2,500 million years ago, when oxygen began to accumulate in the atmosphere of the Earth. So at the end of the Archean and the Great Oxygenation event, we had cyanobacteria, prokaryotes that could carry out photosynthesis, and we had other bacteria that used oxygen to obtain energy. This is more or less the time when endosymbiosis of a photosynthetic cyanobacterium by a proto-eukaryote would have led to eukaryotic algae and later plants, whereas endosymbiosis of an aerobic proteobacterium led to eukaryotic cells with mitochondria. These are the cells that are found not only in fungi and amoebae, but also in all types of multicellular animals.